Triple-negative breast cancer is the most aggressive breast cancer subtype with no targeted therapies and a tendency to rapidly develop resistance to treatment. My research investigates a potential vulnerability in these cancer cells that could help us overcome this resistance. When cancer cells are treated with chemotherapies that block their ability to make new building blocks, they find ways to survive. One key survival mechanism is macropinocytosis. Essentially, the cells drink nutrients from their surrounding environments, including debris from dead neighboring cells. This allows them to keep growing despite treatment. The challenge is our current tools to study macropinocytosis aren't specific enough. The most common chemical inhibitor, EIPA, affects multiple cellular processes, not just macropinocytosis. And previous genetic approaches using short hairpin RNAs have off-target effects that can confound results. Therefore, we need a more specific genetic tool. Karma1 is a protein that regulates macropinocytosis through its interaction with another protein called CAPCB. When Karma1 binds to CAPCB through a specific region called the capping protein interaction motif, it promotes the membrane ruffling that drives macropinocytosis. My hypothesis was that Karma1 and its CPI motif interaction with CAPCB are specifically required for macropinocytosis in 4T1 breast cancer cells. To test this, I use a three-part approach. First, I used CRISPR-Cas9 to knock down endogenous Karma1 and 4T1 cells and measured macropinocytosis using dextrin uptake assays. Second, I rescued the knockdown by adding back wild-type Karma1 to confirm the effects were specific to Karma1 loss. Third, I rescued with a mutant version of Karma1 called Karma1AA that cannot bind to CAPZB to test if it's specifically the loss of the CPI motif that causes the loss of macropinocytosis levels. The results were convincing. Karma1 knockdown reduced macropinocytosis by more than half, and it did not affect cellular proliferation at all, which tells us that the effect is specific to macropinocytosis, not just general cell growth. When I added back wild-type Karma1, macropinocytosis completely recovered to normal levels. This confirms the phenotype is specifically due to the loss of Karma1. But interestingly, when I added back Karma1AA, the mutant that cannot bind to CAPCB, there was no rescue. Macropinocytosis stayed low, just as the knockdown cells. This proves that not just the Karma1 protein we need, but specifically its ability to interact with the CAPZB through the CPI motif. These findings establish Karma1 as a specific genetic tool for studying macropinocytosis in breast cancer models. Unlike EIPA or shRNA approaches, this system allows us to selectively block macropinocytosis without confounding off-target effects. Looking forward, this validated genetic framework opens the door to investigating whether targeting macropinocytosis, specifically the Karma1 CAPZB interaction, could prevent therapeutic resistance in triple-negative breast cancer. Thank you.